Hello, good afternoon, everybody. My name is Daniel Dibaba. From the Tennessee Clinical and Translational Science Institute. Um, we will wait for a couple of minutes just in case other people might join. All right. So I'm Daniel Dibaba from the Tennessee Clinical and Translational Science Institute. Today we'll discuss about secondary data sources for research. Can you all hear me? Good.
Yes.
Thank you. So the objective of this seminar is to discuss about the common source of data for medical research. We will also discuss about the advantages and disadvantages of secondary data source. And we will compare the secondary data source with primary data sources. And we will eventually discuss about the methods to determine if secondary data are sufficient for research. So the source of data for research are primary and secondary. And the primary data are data that are generated by the primary investigators um, from primary sources using survey, questionnaire, interviews, digital devices, and experiments. The secondary data are data that have already been collected by somebody else and are available for research. So secondary data are commonly produced either for routine record keeping by large institutions such as NIH, um, or they are produced for assessment of population health status. So let's take a look at the advantages and disadvantages of the both sources of data. So looking at the primary data source first, um, the advantage is that the searchers can collect the needed data. There is no issue with covariate, the variable that you want to control in your analysis, because you are actively collecting the data, so you can include all the variables in your planning and collect on them from your participant. The other advantage of primary data source is that more complete data with less missing values can be collected. So this might be an issue with secondary data, as we will see, but with primary data, you can reach back to your participant and collect as many data as possible. So there is less data missing. Even with primary data, sometimes there is an issue with data missing, especially if you are conducting a large study. There might be missing values, but um, still you can trace back your participant and collect more data on them if the data are missing. And the study design is determined at the beginning by the investigators in case of the primary data source, which is different in terms of the secondary data source. So the disadvantage of primary data source are that it's very expensive. It takes a lot of time and resource. Um, you have to have some people to collect the data, and it might take a while before you can get the data ready for the analysis. So it's very expensive relatively, and it's time consuming, and feasibility might be an issue um, in terms of data collection for rare outcomes. If the outcome are very rare, or if the outcome um, is a health condition that requires some pathological changes that that takes a lot of years, such as cancer. Primary data source is not a good method to collect the data for such kind of studies. The other one is cost and time that's needed, which can limit the sample size and thereby the statistical power. So primary data source, even though it's a ideal, it can be very expensive, it can be time consuming, and it might not be feasible sometimes. So that's where the secondary data source shines. One is very inexpensive. Um, it has already been collected. It could be an administrative data. It could be data collected for assessment of community health status or the population health status. It's very efficient. Um, so the data has already been collected, and we don't spend time on this. So it's time efficient, and cost-wise also it's very efficient. And it's very appropriate for rare outcomes because the outcome has already happened, and we don't have to wait for years until the outcome are ascertained. The secondary data source improved the statistical power due to the increase in sample size. Since the data has been collected, it doesn't require a lot of resource to include in your analysis. So you can work on large sample size. And most administrative has a lot of participant. There are data on a lot of participant and for long years. So that improved the statistical power. And original research data serves as secondary data for research and meta-analysis as well. So for example, a primary data has been collected and used for one research purpose. We can repurpose the data and expand on investigating the relationship between different factors for secondary aims, to meet secondary aims, or researchers can propose ancillary studies and use the same participant and collected data, additional data, or they can use the same data to answer a different research question than originally proposed. The disadvantage of secondary data is that sometimes the desired data might be missing. So you can't expect to find all the data that you need in your research in secondary data because it might have been collected for a different purpose and you might not get all the data that you need for your analysis and research study. Comparison groups might be missing. So exposure, there might be an issue with the exposure variable or the comparison group in secondary data. That's another disadvantage. The study design cannot be changed. The researcher has to use the study design that has already been used to collect the original data. So the study design cannot be changed. So if the study design was, say, a cross-sectional study where the exposure and outcome have been collected at a snapshot during the same period, the issue is that we don't know which one happened first. But ideally, we want to know that the exposure happened before certain health outcome so that we can say there is a temporal relationship between exposure and outcome. So in terms of secondary data, sometimes you don't get all the desired study design, and you have to use what already exists. The other issue is data accuracy, which might be harder to measure. Because we can't control the data that has already been previously collected by somebody else, and we never know how accurate the data is, but we have to rely on the validation tests that were conducted by the original collectors or investigators. In some other cases, the data might not be current. That means the data was collected in the past, and healthcare has changed since. New medications, new devices might have been introduced, new services might have replaced the old ones. So the findings from those old data might not be relevant to the existing healthcare situation. So that can be a challenge sometimes. So studies on devices and services and data might be available only on the devices and services that's being used, and may not be available on new ones either. So those are some of the challenges with secondary data sources. So at the University of Tennessee, we have quite some data that researchers can use. So among these are the Cerner Health Facts database. The Cerner Health Facts has about data on about 50 million patients and over 300 million encounters from all over the USA. So it's one of the largest databases. Researchers can search online on UTHSC and propose a study, and they can ask the CBMI department to extract data for research. So the other one is the Research Enterprise Data Warehouse. This has data on adults and pediatric patients from hospitals on UT campus. So this is on kind of electronic health record data from hospitals on UT campus, or hospitals that have a relationship with the University of Tennessee. And the TriNet X is the other one. This one provides visual and tabular summary data for the enterprise data warehouse. So you can search for patient populations, and the treatment that you want to investigate. So another source of data is the Serverless epidemiology and end results registry data. So this is abbreviated as SER. So SEER contains data on cancer survival. It contains data from about 19 registries from across the United States. Data are reported, are released on annual basis, but the data lags about 2 years, the release year. So the data can be linked with claims data and Medicare data, and it contains data on cancer incidence, demographics, patient information. treatment, surgical treatment, or radiological treatment, and chemotherapy sometimes, and primary site of cancer, the tumor grade, tumor stage, lymph node, mortality, and cancer survival. So, and also we have information on metastasis as well. To access the SEER data, one needs to complete a request form on the SEER website from the National Institute of Cancer. And there are research database. There are two databases. One is called the research database, and the other is called Research Plus database. So the first one gives a limited access, while the Research Plus database gives a more access, and so it requires an institutional sign up or research proposal to access the full data. So SEER releases data from as early as 1975. And like you said, there is a data release almost every year. And the data lags the release year by about 2 years. So for example, the 2021 release contains up to data from 1975 to 2019 reports. So the SEER data is very big. It contains millions of rows and hundreds of columns. And one can study about the cancer survival and quality of life studies and the likes. You can access the SEER cancer-related data from the seer.cancer.gov. The other data is the National Health and Nutritional Examination Survey, or NHANS. NHANS contains demographic, dietary, and total nutrient intake and supplement intake data. It also contains laboratory and physical examination or other examination data, and we can get the enhanced data from the CDC. Another secondary data that we can use for research is the healthcare cost and utilization projects, or HCUP. So that's found on the Agency for Health Research and Quality. And several data sets can be accessed on the national inpatient data and pediatric data, and there may be fees for data extraction. The data that's found on the healthcare cost and utilization is similar to the Cerner and Tranex data, and can be accessed on millions of patients from across 47 states in the USA. The other secondary data source that we can use for research is the behavioral risk factors surveillance system, or BRFSS. So it's the US premier system of health-related telephone survey and on health-related risk behaviors and chronic health conditions and use of preventive services. It completes about 400,000 adult interviews each year, which makes it the largest continuously conducted survey in the world. So to get the behavioral risk factor surveillance system data, we have to go again to CDC. The National Institute of Health, American Associations of Retired Persons, Diet and Health Study is another second data source. Was designed to improve the understanding of the relationship between diet and health. One can use to investigate, for example, the association between certain nutrient intake or food group intake and health outcome. So one can access the data going to the diet and health.cancer.gov. For genomic data, the MD Anderson Cancer Center from Texas provides genomic data on genomic data commons through the following link. Once we are done with the presentation, we can go and look at some of these secondary data sources links. Another one is a EBI. That's another source for genomic data. So, and UK Biobank is another one. The National Library of Medicine open domain specific data sharing repositories are also found on the NIH, on the National Library of Medicine, .nih.gov. We'll look at these links as well. So two NIH Institute specific repositories are there on the National Heart, Lung, and Blood Institute, BioLink, and the National Diabetes and Digestive and Kidney Disease repository are found on this link. So there are a lot of data to conduct research, a lot of secondary data to conduct research, including on cancer survival. If you want to conduct a research on cancer survival, probably the best one would be looking at the SEER database, and you can access the data without any cost. You just have to fill out a form and request for the data, and you can only you might need to pay something if you want to link it to Medicare or claims data. Otherwise, it's going to be it's mostly free of charge. The HCUP, or the healthcare cost utilization project, probably that one requires cost in terms of data extraction. So we have also some local original research that have been conducted in the past and can serve as secondary source of research data. Among these are the Women Health Initiative and the Look Ahead study, the Sprint study, the Candle study, and Health ABC. We will look at each one of these one after the other. So the Women Health Initiative was is a national study that was conducted here in the U.S. More than 160 postmenopausal women were enrolled in the WHA, and the study started around 1991. It was initiated by the first woman director of the National Institute of Health, and over 68,000 women were randomized to 3 different clinical trials. The first of those is the hormone therapy trial, the diet modification trial, and the calcium and vitamin D trial. Over 93,000 women were enrolled in the observational study. So the observational study was conducted to kind of validate the clinical trial, to confirm the findings from the clinical trial. The findings from this trial has been very impactful and has reduced the risk of breast cancer and improved understanding about whether hormonal therapy can be helpful in preventing certain disease or increasing the risk of another. So to get data from the Women Health Initiative, WHI investigators may also be interested in the scientific interest groups. The scientific interest groups meet by reference conference call regularly to share information, stimulate ideas, and coordinate scientific activities in line with their mutual interests. So the WHI has a lot of specimens, blood specimen, urine samples, and other data. So the blood sample is available from the clinical trials, and the blood were collected from baseline through 9 years, and also from the observational studies. And they include serum, plasma, DNA, and red blood cells, and the like. A lot of specimens are available. So one can go to the WHI website, and there are a lot of information. There are data dictionaries, there are forms, there are ancillary study proposal forms. One can propose study, ancillary study, or one can propose manuscript and request for data. And thousands of research has already been published using the WHI data. So data on wide range of clinical outcomes have been collected, and it's a very good source for research. In addition to the hormone diets and the calcium and vitamin D clinical trials, there are also specimens for genome-wide association studies and biomarker suites of studies. The WHO code studies provide both GWAS data and biomarker data on approximately 22,000 participants, including about 8,500 African Americans, 3,600 Hispanics, and about 10,000 European Americans in the hormonal trial. The WHI, the Human Health Initiative, is also linked with external data sets, including the National DAYS Index and Center for Medicare and Medicaid Services, including parts A and B, and D is said to be coming. And it's also linked with the EPA data for toxicology studies. So researchers can be interested in either proposing a manuscript to analyze existing data, or can propose ancillary study where additional data can be collected from the participant or indirectly from linkage to other data sets through the analysis of WHI biospecimens. So the other study, a very impactful study, is the SPRINT study. It stands for the Systolic Blood Pressure Interventional Trial. At the University of Tennessee, Dr. Karen C. Johnson is one of the main PIs on this study. And the study began in the fall of 2009 and recruited more than 9,300 participants from over 100 medical centers and clinical practices nationally in the US and Puerto Rico. So it's on the systolic blood pressure. It was the largest study that examined whether maintaining systolic blood pressure below the then recommended level would reduce the risk of heart and kidney disease, stroke, and decline in memory and thinking. So the purpose of the SPRINT study is to investigate whether lowering the threshold for hypertension would result in reduced risk of heart and kidney disease, stroke, and decline in memory, such as Alzheimer's. The study was stopped a year earlier than planned for benefit in 2005 to disseminate the results. So the finding was that reducing the original threshold for hypertension from 140 um, systolic over 180 diastolic to 130 reduces the risk of cardiovascular disease, kidney disease, and others. The study resulted in the lowering of the normal systolic blood pressure to below 120 and elevated systolic the elevated systolic to 120 to 129. It used to be above a normal was considered to be below 130, but this was improved based on the result of the SPRINT study. So the definition of a stage 1 hypertension was revised to 130 to 139 millimeter of mercury by American College of Cardiology and American Heart Association based on the SPRINT findings. The SPRINT has a large biorepository with serum, plasma, urine, and DNA. For the women health study, investigators may propose ancillary studies and manuscripts. The website for the SPRINT is here. Another data set, original study that was conducted with UT involved as one of the centers is the Look Ahead. It's an abbreviation for Action for Health in Diabetes. So the purpose of the Look Ahead was to answer whether an intensive life intervention designed to produce weight loss results in decrease in cardiovascular disease or morbidity and mortality in overweight and obese adults with type 2 diabetes. The finding that was published in 2013 is that the intervention did not result in the reduction in the risk of cardiovascular disease, but it improved a lot of the factors in health. So the Look Ahead trial randomized, the Look Ahead randomized control trial began in 2001 and lasted for more than 8 years and recruited about 5,145 participants from across 16 clinical centers in the US. And the diabetes care is provided by the participants' own physicians. The primary outcome were the composite of cardiovascular outcome, including cardiovascular disease-related deaths, non-fatal myocardial infarction, non-fatal stroke, hospitalized angina. So the primary outcome of the composite of these all four conditions. So while the intervention trial improved the overall health by resulting in weight loss, reduced glycated hemoglobin, which means there is improved diabetes control, improved cardiovascular risk factors, it do not result in a significant difference in the primary outcome. So the intervention is one is the intensive lifestyle intervention, and the other one is health education for the diabetic patients, health education and support for the diabetic patients. So eventually, when the study was published in 2013, there was no difference in terms of the composite outcome. But there were some improvements in lipids, in glycated hemoglobin, weight loss, and the likes. Other positive outcomes have been reported in blood pressure, sleep apnea, renal disease, and this was reported by Salvia Meg, one of the authors. So other outcomes from the look ahead study is on cancer, on fracture, peripheral vascular disease, congestive heart failure, and like. So if you want to propose a study, you can look at any of these outcomes in your study. The look ahead study also has a large biorepository of specimen on serum, blood plasma, urine, and DNA. And so one can use for additional study. It also has some bone density scans and data on cognition, physical function, and measures on the subset of the look-ahead study. The investigators may propose ancillary studies, sub-study, or can propose manuscripts. And the bibliography for the published manuscript can be found on the website lookaheadtrial.org. So the candle study is another locally available study, and it is conducted by the University of Tennessee Health Science Center. And the purpose was to examine the separate and combined effects of maternal prenatal actions, the home environment and child life experience, and exposure to potential toxins, the genetic makeup on the child's brain development from birth to about 3 years. So this study is conducted by the University of Tennessee Health Center, and the database is available as a subdomain on the University of Tennessee website. So planning began in January 2005, and it enrolled about 1,503 women by July 2011. The plan was to follow these women until their children were 8 years of age. So the candle study is a kind of birth score study on early factors affecting the neurocognitive development in children. And the population is from Memphis, Shelby County. And the study data includes genetic, environmental, social, and other measures. And the publications can be found on YouTube website as well. The Health ABC study is another study, and has about 3,075 participants on senior aged between 70 and 79 years of age at baseline, and about 45% were African American, self-reported ethnicity. And the data is available from Pittsburgh, Pennsylvania, and from Memphis, Tennessee. And the participants have to be able to walk about 1/4 a mile, or about 10 steps at the baseline at the entry to the study. And the outcome is whether they so the participants are overweight or obese, and so whether frailty or decline in as they advance in age. So what factors can be related to the inability to walk 1/4 of a mile, or about 10 steps. So this is the 10 steps is kind of stairs. So this is designed to assess the relationship of weight and body composition to incident weight-related diseases and disabilities. The collaborating centers are University of Pittsburgh, University of Tennessee, University of California, San Francisco, a laboratory for epidemiology and demography and biometry. So the study began in between 1997 to 1998, and the follow-up was conducted uh, for about 16 years. The overall purpose of the study was to characterize the extent of change in body composition in the older men and women, and to identify clinical conditions accelerating these changes, and to examine the health impact of the changes on strength and endurance, disability, and disease of old age. The data can be accessed on NIH website at healthabc.nia.nih.gov. So that's it for the discussion on examples of source of secondary data. Now let's look at how can we say the data, secondary data sources adequate or sufficient for research. So to determine if a secondary data source is adequate for research, we should examine whether the data set has enough data on exposure and predictors. Um, uh, it should have enough data on outcome. It should have enough data on covariates or factors, additional factors in addition to the exposure and outcome that can affect the relationship between the exposure and outcome. So in order to avoid any spurious association being detected while conducting the data analysis, you want to adjust for certain factors or covariates. And so one has to make sure that your secondary data contains data on covariates. And the other quality metric is about missing values. If there are a lot of missing values, the quality of the data might decrease. So we have to make sure that there is a minimum missing values in the secondary data source. And the other is availability of data dictionary and code book. So the data dictionary defined how data are derived, what data, for example, what a column refers to, what a given name and columns refer to, and what are the units for each of the measurements and the like. So it provides a metadata about the data that we are going to use for our analysis, and it's very helpful for the correct interpretation of the result and for correctly conducting the data analysis. So data dictionary is always very important and should accompany the data. Most secondary data sources publish their data dictionary, and also major research studies publish their data dictionary along with their research, and they are usually publicly available and can be downloaded and used while conducting data extraction and data analysis. Another metric for the quality of secondary data is attached to be validated. If the data has been validated for measurement accuracy, this is another metric, and usually we have to look for this. Or if the data has not been validated, we have to state that the data has not been validated. So to recap, secondary data are used for research on utilization of health services by special populations. Ancillary studies, the secondary data can be used for conducting ancillary studies to main study using biorepository. And secondary data can be used to study use and cost of hospital services for study on medical practice variation, comparing two different um, procedures. For example, for certain surgical procedure, maybe an open surgery can be conducted, or laparoscopy can be used for conducting the surgery. So you want to compare the outcome of these two in terms of readmission, in terms of recurrence, treatment failure, infection, and the likes. So secondary data sources can be used for those purposes, for comparing health practices. And it can be used for research on quality of care, on research for the impact of health policy changes. So secondary data sources can be used for that. So for example, the enhance collects data on nutrition, and some policies have been passed concerning, for example, the use of saturated fat, and that has most likely changed the availability of saturated fat or labeling of saturated fat in the supermarket. And if you are interested in looking at whether saturated fat intake and unsaturated fat intake has an impact on cardiovascular health, you can conduct a research based on the enhanced data. And so it can be used for assessing the impact of health policy changes. It can be conducted for assessing the access to healthcare as well, and for diffusion of medical technology. Um, the secondary data source can be used for research on association between predictor and health outcomes. It can also be used for study of causal relationships, especially for biorepositories and genetic data. So generally, there are primary and secondary data sources for research. Um, One can contact the study investigators and collaborate with them for a better understanding of the study and existing data sources. And there are abundant secondary data sources that can be used for research purposes. Like we said, secondary data sources are relatively inexpensive and very efficient, less time consuming. So it can be used for research purposes. So it's a good idea to consider secondary data sources for your next research. Secondary data sources are efficient in terms of time and cost, but may not have all variables needed for research, so you have to consider about that as well. The time and cost constraints limit the statistical power of primary data source for research, where the second data source shines in terms of improving the statistical power. So that's a last, we can go back and look at some of the links to the beginning from Ser. So this is a website for SEER. So like we said, the recent data, the recent release has incidence data ranging from 1975 to 2018. Usually it's released around this time. So I think, I'm not sure if we have the release for 2021 yet. So we have two databases, the research database, and this has some kind of limited access. And then the research plus database, this has more complete access, but it requires institutional sign up or proposal for the research study or manuscript.
Daniel, we're not able to see the screen. We're seeing your slide.
Oh, sorry. Let me share the new one. Yeah.
Thank you.
You're welcome. So SEER is one of the very large databases, and it's produced and it's released annually by the National Cancer Institute. One category the data, and it's very easy to access. It has millions of rows and hundreds of columns, so you can conduct research on cancer survival, and you can look at whether certain treatments have the desired effect on cancer survival. So in terms of outcome, there is data on primary sites as well as on metastasis.
I had a question, Daniel. Is it okay if I ask?
Yeah, sure.
Is it possible to get access to the local registry, for example, to the registry at Tennessee? So, because SEER has 17 sites.
Yeah.
So is it, I know that there's a separate registry that you can get access for the California registry.
Yeah.
If people have used that database, and the variables are different, and how they present data is different as well compared to SEER. But is there one for Tennessee?
What I can say is that it's possible to get states like a state registry data from a state registry, so you can limit your participant into only the state of Tennessee. In terms of whether you can directly communicate and get the data from the local registry is not something that I've previously considered, but I believe it's possible.
And what is your thoughts and experience about using Flatiron or some of the commercial large set databases? Can you speak about that, if you don't mind?
What is that again?
Like there are some other commercial large electronic healthcare databases.
Mm-hmm.
I'm talking from oncology perspective.
Yeah.
So like SEER Medicare, it's requested on permission, and one can also request permission to get commercial Medicare database as well, for
Right.
private insurance. Instead of
Right.
Medicare, you can get private insurance.
Yeah, so those ones, relative to the already administrative data that you can freely access, if you want to link the SEER database to additional claims data or Medicare and the likes, treatment and the likes, or health insurance claims, or probably pharmacy and the likes, it's gonna cost money, but again, relative to conducting an original primary study, it's relatively cost effective.
Thank you. And then is there a statistician who's well versed in this that we can use on studies moving forward?
Yeah, so I personally use SEER data about twice or three times. I've accessed and downloaded the data and used. One of the things that I looked at was whether the different surgical procedures would result in a better colorectal cancer survival. Personally, I've worked with SEER data, but also I think a lot of our faculties and biostatisticians are familiar with it.
Okay, thank you. I'm new to University of Tennessee, so that was helpful.
Yeah, thank you.
Thank you.
Is there any question? Any other question? So
I had another question. In terms of getting genomic data, you said that there was a database that had genomic data that was through. That was sorry.
MD Anderson.
Right, the GWAS or the Common Genomic or their pipeline, the GDC.
Yeah.
So is that free as well?
Yeah. Let's look at next to this one. That is available online, and some of the data have already been used for other publications. You can download the data yourself. Yeah, it's freely available. I think in terms of UK Biobank, I think they might request for some payment. Probably, I don't know if this for only for processing charges. You have to sign up and then wait until they give you access to the database, and then you have to, it has a waiting time. In terms of the MD Anderson Cancer Center, it's freely available, and we can look at what it looks like, and also other databases.
Thank you.
You're welcome. So this is the CDC or website for the enhance. So we can access the data on, we can request for the data based on the years the survey was conducted. So one can, for example, if we click on this, so we have the data documentation and core books or data dictionary. And I think somewhere we can also download the data. So, yeah. So let's look at the genomic data, right? Let's go to the genomic. Okay. So as you can see, there are several studies that have been conducted, and as you can see, there are several studies that have been conducted, and there are certain data and also results. You can also view the results like here. I'm not an expert in genetic data analysis, but this is, I think, it's PCAs, like kind of clusters. And so you can download the data and use it in your own research. And the other one is,
What software would you use to review genomic data like that?
Once you download it, you can use R or SAS or any other software. Like, so for example, you can use the bioconductor packages, R packages to look at the data and conduct analysis. You can also use, some people use peeling. I don't know if you are familiar with that for the data analysis aspect.
Can they do can it be done on SPSS or a SAS?
I'm sure SAS is a very it's a very advanced data analysis tool. So I think it has a lot of genetic data analysis resources. But I myself, I'm not an expert in genetic data analysis, but I have used R in the past. I've also used peeling.
Thank you again, Daniel. Thank you.
You're welcome.
What's that last resource you said? Did you say peeling? Unpeeling?
Peeling. Yeah, let me go to, let me stop sharing this, and we can see link to yeah. So do you see this? Just searching on Google, there is a coggenomics.org. Can you see it? So
Yes.
it can be downloaded and used, and there is a Google group that helps you with how to like write your codes if you run into any bugs while conducting data analysis. You can conduct GWAS study. You can conduct the quality controls and the likes. And it's a good tool. It's a free open source software, and Christopher Chang almost responds to requests if you are conducting the analysis yourself almost on a daily basis on the Google group. So that's it. That's all on secondary data source as source of data for research. I think you enjoyed it, and probably you have an idea of which one to use for your next research. If you have any question, you can send me an email at dbaba@uthsc. If you want a consultation and support for research design or data analysis, you can also go to our website, the tnctsi.uthsc.edu. Thank you.
Thank you.